Thank you, Hopi, for that really nice introduction. It's really, I want to particularly thank the organizers for putting on this meeting. It's such a great thing to have this standalone meeting for population, evolutionary, and quantitative genetics, which has been such a fundamental part of the Genetic Society of America for the last century. And so to have our own meeting is really amazing. And also to bring together such a nice community of people. There's so many people here that I know and love, and I'm looking forward to talking to people more. And it's also a great honor to be asked to give this talk. So thank you very, very much. Okay. So research in my lab is really focused on three main areas. We'd really like to understand the genetic and the genomic factors that underlie the processes of adaptation and speciation. And that's really what I'm going to focus my talk on today. I'm going to tell you kind of globally what we've learned from since the time that I was a postdoc at Stanford using this model system. And then I'm going to delve into a kind of a story that's been going on in my lab that's still ongoing. But we've also been long interested in understanding the genetic and neural mechanisms that underlie behavioral evolution. And I'm also very interested in understanding how the genome itself evolves and how this evolution of the genome contributes to the processes of adaptation and speciation. And we particularly study this on the sex chromosomes, and we've also started studying the centromeres and how they evolve. So I'm happy to talk to people about these other projects. But let's turn to the focus of the talk for today, and that is understanding this question of how is it that organisms adapt to new environments. And so if you're, for example, a small fish, you can be found in small, slow-moving streams, large, deep lakes, or the ocean. And in each of these different habitats, you encounter different predators, different prey, different parasites, different abiotic factors. And so we'd really like to understand how is it that organisms can live in these very different habitats. Now, as a geneticist, what most of us here want to understand is the genetic basis of this adaptation. And so we'd like to understand the connections between genetic variation and phenotypic variation. And in particular, we're interested in those phenotypes that are subject to natural or sexual selection. And then for evolution to happen in these traits, of course, there needs to be an underlying genetic basis. So we'd like to understand the genetic changes that contribute to evolutionary changes in these phenotypes. Now, the specific questions that I've really been thinking about for a long time now is, this classic question, which Jonathan's touched on a little bit on last night, is does adaptation occur via few mutations, a very large phenotypic effect, or many, many mutations of very small effect? I'm also interested in understanding whether things like genetic linkage or pleiotropy might facilitate rapid adaptation to different environments. And ultimately, as my developmental genetics background leads me to want to know what are the actual genes that contribute to adaptation. Specifically, we'd like to understand whether coding mutations or regulatory mutations are more likely to contribute to adaptation. When the same phenotype evolves, is it because there's different genetic mechanisms used or the same genetic mechanisms? And if it is the same genes, is it due to new mutation or standing variation in the ancestral population? Okay, so we study these questions in this small fish that Hopi introduced you to, the three-spine stickleback. Just a little bit more information about why I think they're such a great model system. So these are small teleost fish. They're just a few inches long. And they live in all of these different habitats. So they live in both marine and freshwater, marine habitats as well as freshwater lakes and streams. And it's really in these freshwater habitats that we see extensive phenotypic variation. So pretty much all stickleback populations vary in behavior, morphology, physiology, and life history. We also have this really great set of evolutionary experiments. So there's replicate evolutionary events. So sticklebacks have invaded freshwater from marine habitats thousands and thousands of times over in natural populations. Now, what I love as a geneticist is that I can take these very divergent populations, I can bring them back to the lab, I can cross them, and I can get perfectly viable and fertile offspring. And this really allows us to take a genetic approach to trying to understand what the genes are that control these differences in phenotype. Now, for sticklebacks, we have a lot of genetic tools. And now it seems really easy to sequence a genome. Back when I started working on sticklebacks, we didn't have those sorts of resources. But really, we have kind of a lot of the tools that are available to more traditional model systems now available in sticklebacks. So just a little bit more about the evolutionary history of the species. So the ancestral form of sticklebacks, we think, was a marine form. They were swimming around in the oceans, and about 10 to 15,000 years ago, when glacial ice that covered most of the northern hemisphere began to melt, this led to the creation of many new freshwater lakes and streams, and marine sticklebacks were able to invade these newly created lakes and streams. And again, today, when we look at these freshwater populations, we see this enormous diversity, again, in morphology, physiology, and behavior. Just a little bit more about some of the specific phenotypes. So this is just a comparison between marine and freshwater sticklebacks. So these are male sticklebacks tending their nests. They have these very elaborate courtship dances that they do to attract females to their nests, and they get these beautiful red throats and blue eyes. So marine sticklebacks are always very heavily armored. So these are stained with alizarin red to highlight the bone. And marine sticklebacks have this set of spines on their back, spines on their belly, and then these bony plates along their side. Whereas most freshwater sticklebacks have lost these posterior bony plates and just have these plates here in the anterior. And they often also have loss of either dorsal spines or the pelvic spines. So these differences in the plate phenotypes and armor phenotypes are really dramatic and something that people have focused on for a long time. The skeletal phenotypes is what attracted me and David to this system. But there's a lot more differences between marine and freshwater sticklebacks. So there's differences in size, in this pigmentation. So marine sticklebacks tend to be very silvery. These freshwater sticklebacks tend to be more striped. There's behavioral differences. So marine sticklebacks actually move into freshwater to breed. Babies are born in freshwater and then move back out into the saltwater for the rest of their life, whereas stream resident and freshwater resident sticklebacks live their whole life in freshwater. They do not migrate and, in fact, have lost the ability to tolerate saltwater, so they will die in saltwater. There's also life history differences between them. Okay, so there's just this great amount of diversity and all sorts of phenotypes. And so how do we actually get at the genetic basis of this? Well, this is an approach familiar to many of you in the audience, which is basically quantitative trait locus or QTL mapping. We essentially start with fish from populations that look very different from each other, behave differently from each other. We cross them in the lab. We make F1 hybrids. We intercross those F1s. We make F2s. And then we phenotype those F2s for whatever traits we're interested in, skeletal traits, behavioral traits, and then genotype those F2 individuals with markers that are distributed across the genome. Nowadays, we practically just sequence the genomes. Didn't always do it that way, but and then you look for associations between genotype and phenotype. And then you can ask, well, what are the genes and the regions that are associated with those phenotypes? And the ultimate goal is to get to identifying the molecular changes in those genes that were important for the phenotypes that we're interested in. Now, kind of touching on what Hopi said, when I first started working on sticklebacks, and we had this epiphany that this would be such a great model system because of all this diversity, you could cross them. This was back in the dark ages. Back here in 1998, if you went to GenBank and asked how many entries were there for threespine stickleback, there was, a few hundred. So David and I developed the first genetic linkage map, microsatellite markers for stickleback, which allowed us to kind of proceed with the genetic mapping. And then this facilitated the development of additional resources, including a whole genome sequencing project that was completed in 2005. It was a Sanger sequenced genome. This is before any sort of next generation sequencing. So we've come a long way in terms of the development of genomic resources and a community of scientists who have studied these fish from many different perspectives. So I recently had the opportunity to kind of ask, well, what have we actually learned from these genetic studies in the past 15 years or so since the development of the first genetic linkage map? And to do this, I teamed up with David Marquez, who was a PhD student with Ole Seehausen at the University of Bern. We actually did this before I moved to Bern, but David had basically made a database of all the QTL mapping studies that have been done in Sticklebacks. So there were nearly 30 in that 15-year span. And there were over 1,000 QTL that had been identified. And so we kind of classified these into different categories of traits. So there's a lot of morphological traits, but we categorize these as things related to feeding or defense or shape, body shape, also some traits related to swimming or pigmentation. There was also a few traits related to reproduction, and then a class of behavioral traits. Okay, so we took this big database of QTL, and then we applied them to the questions that I posed at the beginning of my talk. So does adaptation occur via few mutations of large effect or many of small effect? Does linkage or pleiotropy facilitate adaptation? And then what are the actual genes that underlie adaptation? Now I have the word adaptation here kind of highlighted in orange, because really I have to say that we're studying phenotypic evolution. So this is a caveat of our analyses where these are the genetic basis of phenotypes. We think that the phenotypes that we've mostly studied are adaptive, but we don't always know that. And so just keep this caveat in mind that we're talking about the genetics of phenotypic evolution, but we think that we can say something about the genetics of adaptation from what we've learned. Okay, so this first question, does phenotypic evolution occur via few mutations of large effect or many mutations of small effect? This is a long going debate. And Fisher, of course, proposed the infinitesimal model, which stated that all of adaptation is many, many mutations of very, very small effect. And there's really no point in looking for those mutations because there's so many of them and you'll never find them. Now, this was later modified by Muthu Kimura, who recognized that if a mutation did have a large effect, it was more likely to fix. And so that was one modification. Then Alan Orr later developed this theory to suggest that you might expect to find a few mutations of large effect and many mutations of relatively small effect. So we wanted to ask this in our data set. So this is plotting all of the QTL that have been found. So percent variance explained is just how much of the variance in the phenotype is explained by a particular QTL. 100% is essentially a Mendelian QTL or locus, and things that are down here in the 0 to 20% are relatively small effect. So we see what was predicted by Orr, that there's a few things that have relatively large effects, and then lots of things that have relatively small effects. There's lots of caveats of these sorts of analyses. We're certainly not detecting all the stuff way down here that have very small effects on phenotypes. But I think the important point is that there is a distribution of effect sizes with these few things of large effect and lots of smaller effect. Now we wanted to ask whether this was driven by particular types of traits. And so we broke into our different categories, and as qualitatively, these distributions look quite similar. So we see a few more things of large effect for these defense traits, but for the most part, we see a similar distribution. I want to point out one category here, which is close to my heart, which is behavioral traits. So the distribution for behavioral traits is not much different than for morphological traits. We've worked a lot on the genetics of behavior in my group, and people told me that that was crazy, that we would never be able to identify QTL or loci that were important for behavior because it was all going to be things of small effect and environmental. But I think this is a demonstration that it's not really a fundamentally different genetic architecture for behavior and morphology. The caveat again being it's a relatively small sample size. Okay. So the second question is this question about linkage and pleiotropy. So if you think about an organism adapting to these different environments, there's of course multi-dimensional kind of selection happening to parasites, predators, abiotic factors. And so it's been proposed that things like genetic linkage or pleiotropy, where multiple traits are inherited together, might facilitate rapid adaptation or phenotypic evolution. So we wanted to look at this a little bit more closely in the data. So we looked at the distribution of QTL across the genome. So this is the chromosomes of stickleback, they're 21. And then again, kind of where you see lots of bars here is just represents the number of QTL that have been mapped to a particular chromosome. And first we'll focus on all of the QTL together. There are 5 chromosomes for which we find more QTL than you would expect, given either the chromosome size or the number of genes on that chromosome. One of these is associated with a known inversion between marine and freshwater sticklebacks. That's on chromosome 21. The rest are not associated with any sort of known rearrangements between marine and freshwater sticklebacks. The other thing I think is really interesting is that if you start breaking down into these different categories, you find that, for example, chromosomes 4 and 21 are always associated with clusters of traits, no matter what the category. But then there's this cluster, say, on 20, which seems to be associated with feeding sorts of morphologies, this cluster on 16, which is associated with shape, and this cluster on 7 associated with defensive traits. And I'm going to come back to this question for the second half of my talk of what might be causing this clustering of QTL. But briefly, I just wanted to say what we've learned about the genes that contribute to phenotypic evolution and addressing these questions. So thus far, there's a relatively short list of genes that have been associated with phenotypes in sticklebacks, and I want to say that most of this work was done either in David Kingsley's lab at Stanford or Craig Miller's lab at UC Berkeley. I'll also say it was Craig Miller's poster when he was a PhD student on zebrafish jaw development that actually made me start working on sticklebacks. But then we got Craig into the stickleback world, so everything, which is great. And in fact, I think Craig's lab has a few more stories coming out, really beautiful work that will add to this list of genes. But okay, so this is a relatively short list of genes. What can we actually say about them? Well, most of the genes identified so far are genes that we know and love from developmental genetics. So they're genes that are involved in development in model organisms. And so the mutations identified so far have been regulatory. So they're changing the regulation of these genes in specific times or specific places, and thus avoiding any sort of negative pleiotropic consequences that occur if you just knocked the gene completely out. This is just a small handful, so it could also be, of course, that coding mutations are important for stickleback evolution, but we haven't identified any so far. When the same phenotype evolves, are the same or different genes involved? About half of the time you find that the same genes are involved, and about half the time you find they're different. So whether you think that's a lot or a little, depends upon if you're a glass half empty or half full. I think it's a lot. I will also say that recent work in my lab suggests that this may not be true for all population comparisons that we look at, and I'm really interested in why some sets of populations might show more kind of convergent or parallel evolution than others. But when it is the same genes involved, is it because of new mutation or selection on standing variation? Well, selection on standing variation is really common and I think contributes a lot to the rapid evolution that we see in sticklebacks. But Frank Chan, who gave a talk earlier, also used to work on stickleback, and actually has a beautiful story showing that repeated mutation at this PIDX1 locus leads to independent losses of the pelvic spines in multiple freshwater populations. So both are happening in sticklebacks, and the extent to which standing variation versus new mutation contributes is still an open question, I think, in the field. Okay, so now I want to move from this kind of general lessons in stickleback to talking more about a specific story. And this is really focused on this observation that QTL seem to be clustered in the genome. And so one of the big questions, I think, for many of us who do QTL mapping, genetic mapping, is when you find these clusters where lots of traits are mapping to the same place, is this due to pleiotropy or linkage? And this is a paper or a sentence that's standard in every paper on this topic. It could be pleiotropy, it could be linkage. We don't know. Well, we'd like to figure that out. So just to make sure the definition is clear, so pleiotropy would be if a single mutation controls multiple phenotypes. My student Sophie refers to this as a master mutation. If it's linkage, it's multiple mutations that are tightly linked to each other, that are inherited together, each independently controlling different phenotypes. And Sophie calls this a mass of mutations. Okay, so we've been trying to delve into this question. Diana Renison, who's a postdoc in my group, has been taking a kind of genome-wide approach to this question. And really, she's focused on asking, does pleiotropy constrain or promote adaptation? Her preliminary results suggest that, that there's kind of a Goldilocks effect. Too much pleiotropy isn't good. Not enough pleiotropy isn't good. And so a little bit of pleiotropy seems to be involved in loci that are important for adaptation. But I'm happy to, I won't have time to go into Diana's stuff, but I'm happy to talk to people afterwards. Instead, I'm going to focus on the work on my PhD student, Sophie Archambault, who's really a phenomenal student who followed me from Seattle to Bern. So Sophie's been focusing on this QTL cluster on chromosome 4. This cluster is large. There's lots of traits that map there. It overlaps with a region of genomic divergence on chromosome 4. So Felicity Jones did a beautiful work sequencing the genomes of marine and freshwater sticklebacks from around the world and found about 80 regions of the genome that are repeatedly diverging between marine and freshwater sticklebacks. And one of these is on chromosome 4. And so basically there's this region of about 16 kilobases where we see fixed differences between marine and freshwater sticklebacks, and that's true of sticklebacks from around the world. So we've really been honing in on this particular region of the genome. So it's 16 kilobases, and it contains 3 protein coding genes, a gene called ectodysplasin, or ETA, and then these 2 other genes, TNSF13B and GARP. So those 2 genes on the right are, we don't know a lot about them. In humans, they're suggested to be important for immune cell development. We do know a lot about ETA, and I'm going to really be focusing on that. It's a secreted signaling molecule that's involved in the development of ectodermally derived tissues across vertebrates. So across this 16 kilobases, there are also 19 fixed polymorphisms between marine and freshwater sticklebacks. So there's both SNPs and indels. And so again, if you sequence freshwater marine sticklebacks, you'll find that they have alternate alleles at these 19 different polymorphisms. Now, we know that the ETA gene itself is responsible for 3 phenotypes that have been mapped to this region of the genome. So again, this is a lot of work both from David Kingsley's lab and then work from my own lab. We know that the marine version of ETA is expressed at a higher level than the freshwater allele. Transgenic work, so basically simply overexpressing the ETA gene under the control of a promoter that just drives expression throughout the body, causes a freshwater stickleback. So if we overexpress ETA in a freshwater stickleback, we can put plates back on the stickleback. It also affects this lateral line phenotype. So the lateral line is a neurosensory system that's unique to aquatic vertebrates. And freshwater sticklebacks have this kind of single line of these hair cells that are found on the surface of the fish, whereas marine sticklebacks have plates, and where the plates are, you see kind of this dorsal ventral arrangement of these sensory hair cells. So we also know, again, from our transgenic overexpression studies, that if we overexpress ETA, we can put this marine lateral line phenotype on a freshwater stickleback. Work from my lab also showed that if we overexpress ETA, that it also causes differences in schooling behavior. So marine sticklebacks school very strongly, which is thought to be a protection against predators. And when they do school, they swim in a very parallel body position with this model system, that little model sticklebacks that I don't have time to go into. But freshwater sticklebacks tend to not school, and if they do school, they really swim at an angle to the models. And so it's the ETA gene actually controls variation in how well the sticklebacks can school, which we think is probably related to this lateral line phenotype. And I also want to highlight that this is one of my favorite papers from my lab, and we published it in Genetics. Published there, it was one of the most pleasant publishing experiences I've ever had. So that's my plug. Okay, so we know that this ETA gene has these pleiotropic effects on these 3 different phenotypes, but what we don't know is it due to the effects of a single mutation of these kind of 19 potential changes across the haplotype? Is one mutation affecting all 3 phenotypes? Or is it a linked set of mutations that are affecting each of these phenotypes independently? And so our approach is really to do finer recombination mapping to try and break up the associations across this small haplotype, and then do some functional experiments. The problem is 16 kb is a really, really tiny region of the genome, and we would have to, I have a beautiful fish room in Bern, but that would be a lot of fish tanks and a lot of crosses to try and really narrow down this region in the lab. So we turned to an approach that we've heard already a bunch about, which is association mapping, in a population that is in Seattle called Lake Washington. So my old office is kind of like right here on Lake Union. And in Lake Washington, we actually found that there's a polymorphic population. So the fish are in freshwater, but they're variable for this plate locus. So a lot of fish are completely plated. So they have the two marine alleles at the ETA locus, or we call the CC complete. There's also low-plated fish, which have the freshwater alleles at this locus, or the low alleles, and then partially plated fish tend to be heterozygotes at this locus. So this population, as far as we can tell, is freely interbreeding, so there's not a lot of any sort of background structure, but they're polymorphic at this locus. And so we wanted to use this population to do association mapping. So again, we have our 2 different phenotypes, really 3 phenotypes. And then the idea was that maybe in this natural population, there's been recombination across this interval that would break up these haplotype markers. And so the idea is that if we could go out into the wild and sample these fish, that we might be able to find the SNPs across this region, or polymorphisms, that were most strongly associated with variation in the plate phenotypes. So that is what we did. We sampled close to 1,000 fish in 2 different seasons, and then Sophie genotyped these fish with markers across the haplotype, and then we phenotyped these fish for all of the traits that map to this kind of big QTL cluster. So there's tons of traits that have been mapped to this big region of chromosome 4. We wanted to know if there were any other traits that actually map to the ETA locus. So the first important result is that we actually found recombination across this haplotype. So this is, again, the cartoon of the haplotype. Most of the fish did not show any recombination. So if the fish was homozygous complete at this marker, it was homozygous complete at all of the markers across the haplotype. But we did find about 1/3 of the fish showed evidence for recombination across this haplotype, so that it was, for example, homozygous complete at this marker, and then a heterozygous at all of the markers along here. This is a little bit surprising. In lab crosses, this region of the genome actually shows almost no recombination. So the fact that we're seeing about 1/3 of the fish are recombinant across this interval, to me, is really quite interesting. So then we needed to phenotype all these fish. Someone mentioned earlier, phenotyping is probably the hardest thing to do now, or the most time-consuming. We had a really great bachelor's student, Louis Berchi, who took this on for his bachelor's thesis, and he phenotyped all of the morphological traits that we know map to this kind of larger region on chromosome 4. Okay, so what did we find? So our current results are that the plate number trait maps most strongly to these kind of two indels, 3 and 4. These loci explain about 71% of the variance in the phenotype, which is consistent with previous genetic mapping studies. Of course, the associations are extremely high across this whole region. I mean, these lot scores are pretty crazy. But it's because the whole region is associated, but these are the strongest associations. So the other markers are recombining away. Okay, so what about other phenotypes? Do we see that they map to the same or to different SNPs? Well, the Neuromast pattern looks pretty much exactly like the plate pattern, where we see the strongest associations here at the indels 3 and 4. They explain about 65% of the variation in the phenotype, which again is consistent with our previous linkage mapping results. Only one other trait actually mapped to this region, and that was the length of the pelvic spines. It actually maps slightly differently. The percent variance explained here is quite small. We have a lot of power just because of the number of fish. It's hard to see our genome-wide threshold here is right about a lot of 3. So this is a significant, statistically significant association. So what we found is basically a few, but not all traits that map to this larger region of chromosome 4 map to the 16 kb haplotype. So really the two phenotypes, we didn't measure schooling in these fish, the bony plates and the Neuromast number map to this haplotype. There's also this weak association with pelvic spine links, but these other traits must map to elsewhere on chromosome 4. To answer our question, though, we find that it's really mostly pleiotropy. So basically the same genetic, probably the same SNPs are controlling these, are controlling both the plates and the Neuromast. But then there's maybe a little bit of linkage with this pelvic spine phenotype. So all of this really now begs the question is, what is the actual mutation? Is it one of these SNPs, or is it something that we don't know about? And so we've been delving, this is where we were really the recent kind of data in the lab. We've been delving into this more closely. So to do this, it's a little bit more complicated than I told you before, because in our previous crosses, I want to point out that fish that are homozygous marine, big A, or heterozygous can be completely plated. So there's some variation due to modifiers of whether a heterozygous fish is complete or partial. And so we really wanted to focus on the low fish, because the genotype-phenotype association is much stronger. So for example, in this particular cross, 93% of fish that were the freshwater allele little a were low plated. And that was true, again, in a second cross. And probably the reason this isn't a little bit higher is because these markers were not perfect. They were recombining with the actual causative locus. So in our data, if we only look at the low-plated fish, we find that almost 98% of them that are low-plated are low, or freshwater at this locus. And then there's a small percentage of these 2% that are heterozygous. So this kind of gives us a baseline for what we think the causative mutation might be. So now we wanted to look in our recombinant fish and ask, was there kind of, where do we see the strongest association, and is it approaching this 98%? So I'm going to show you some kind of raw data here. These are basically Sophie's recombinant fish, which are low, and these kind of lighter purple, and then heterozygotes here. So these are all the very low-plated fish that are recombinant. And if you just focus on these few fish here, so this SNP, or this polymorphism, indel, shows the strongest association. It's 87.5%, and then it kind of falls off at the 2 markers on the either side. So we're really interested in these kind of set of 12 fish where we've really honed in on these recombination events. So I think you can see most clearly here that basically these fish tend to be our low at this marker. They're heterozygous at this marker, and then there's these fish that are low at this marker and heterozygous at this marker. And so either this polymorphism indel 3 is causative, and it's just not a perfect association, or there's something kind of in between these 2 markers, which are 600 base pairs away from each other. And so Sophie is literally, as we speak, sequencing these 12 recombinants. She sent me an email wishing me luck in my talk, and I was hoping it would have the data, but it didn't. So, but essentially, my intuition is that this indel 3 might actually be causative, and so that what we're doing now is CRISPR-Cas9. So Matt Josephson is a postdoc in my lab, and he's really getting this system working in sticklebacks. We can knock genes out. That we can do quite easily. But for this project, what we really want to do is these allele swaps. So put the marine version of whatever what we think the causative change is on a freshwater background, and vice versa. So hopefully we'll make some progress on that soon. Okay, so just in the last couple of slides, I want to summarize and give you also a sense of what else we're doing. So far, I think I've shown you that we think pleiotropic effects of ETA on these multiple phenotypes. But what we don't know then is whether this locus, this entire 16 kb, has effects on other phenotypes that we don't know about. I'm really intrigued by these 2 genes that are next door that seem to be related to immune function. We know that the parasites differ between marine and freshwater sticklebacks, and it could be a strong source of selection as well. So that's something I'm afraid of immunology, but I'm gonna think we'll start thinking about that a little bit more. And then really the big question still is, what is driving selection on this locus? So there's incredibly strong selection for this freshwater haplotype as soon as the fish move into freshwater. And that's illustrated here, where we know that marine sticklebacks move into freshwater. There is some low level of hybridization with stream resident sticklebacks. And so marine sticklebacks carry this freshwater haplotype at anywhere from 1 to 4% frequency. That's the heterozygote frequency. And so, but then the stream resident fish have it at this low plated phenotype, or freshwater haplotype is basically fixed. So what might be driving this selection? It's very strong and very rapid. So work from Mike Bell at Stony Brook has shown in this lake in Alaska where all the sticklebacks were basically killed. And marine sticklebacks invaded this freshwater lake. So in 1990 or before 1990, there were no sticklebacks in this freshwater lake. Mike went in 1990, he found a bunch of sticklebacks. They were all completely plated, suggesting that they were marines that had gotten into this freshwater lake. And within the course of 20 generations, there was almost complete fixation of this freshwater haplotype, as measured by the complete fixation or nearly complete fixation of this low plated phenotype. So this is really strong selection over a short amount of time that was facilitated by the standing variation that was present in the marines. And so we are very interested in trying to understand what's driving this. Currently, Sophie is testing the hypothesis that low levels of phosphorus in the diets of freshwater sticklebacks might be important for adaptation. So phosphorus is a major component of bone. It's also a major component of ATP and RNA and DNA. And so the idea is that maybe in freshwater, where phosphorus is very limited in the diet, that sticklebacks lose some bone in order to put phosphorus more into growth in these nutrient-limited conditions. So we have a huge experiment going on right now with marine and freshwater sticklebacks and low phosphorus and high phosphorus diets, and variation at the ETA locus to try and test this hypothesis. So I can tell you about that in a in another year or so. Okay, but with that, I would like to stop and just thank really my collaborators, Sophie, who's a terrific PhD student, Lewis, who worked really hard on the phenotyping, Matt has been getting the CRISPRs going in the lab, and then Hillary and Verena are two technicians in my lab who kind of make everything work. Our collaborators at the University of Washington, Dave Beauchamp and his students who helped us with the collections from Lake Washington. And I also want to just acknowledge David Kingsley and Dolph Schluter, my very long-term collaborators and friends who really helped we together kind of develop the stickleback system, which I still think is a supermodel. So, and with that, I will be happy to answer questions.